Good day and welcome to this channel, Callista's Bio Class. Now, I am here today to give you a very important information on the Neco specimen, the Neco exam for this year 2024. I've talked about in detail specimen A, B, C, their similarities and some of their differences. Now, I want to talk about another important specimen, specimen D and specimen E. Remember that specimen D is a potted bean seedling, and specimen E is a potted Guinea corn seedling. What does this mean? It means that the teacher has to throw a bean seed and a Guinea corn seed into a soil that is in a pot, and make sure that this this thing grows, the bean seed and the Guinea corn seed grows for at least 1 week, which is approximately 7 days. Now, once this has grown, of course, all conditions for germination should be present: sunlight, good soil, viable seed, water. Now, and of course, air, temperature, air, that is oxygen. Now, that being said, I want us to look at a very important thing about this too. They would most likely ask you, because they know that they are growing a 1 week old seedling. For the beans, which is a dicot, most dicots, when they germinate, they undergo epigeal germination. That is, by 1 week old, the bean seedling has not come out of the soil. It is still under the soil, or inside the soil rather, while the maize seedling, by 1 week old, which is approximately 7 days, which is still the same thing as 7 days, the Guinea corn seedling plant has started coming above the soil, and you will see it very well. That being said, you know that dicot, which is a beans has an epigeal germination. Now they want you to compare. Now, because you know that the beans is inside the soil, you can't see it, but there will be a pot labeled specimen D, and the bean seed is inside. Now, they may ask you to gently or carefully remove the seedling that is inside the potted plant that is labeled D. You get, and examine it with the one that is in the potted plant labeled E, which is the Guinea corn. Now, you need to know some things. That when a maize, when a Guinea corn, which is a monocot, and a beans, which is a dicot, when they are growing, there are some features you see in the maize you don't see in the dicot, which is the beans. So these are the features I want us to look at and note as observable features, which they will most likely ask you, and also know their functions. You get? So let's go. Watching this video, you get a paper, you draw in a tabular form, you draw rows. You have 1, 2, 3, 4 columns. So you're having a tabular form, and they're having 4. That is, columns run from top to bottom. That is what I mean by columns. Now, the first column, you're going to write specimen D. Now, remember, we're trying to look at those things that we can see in the young bean seedling that is present there while it's germinating, while it's growing. And the things you cannot see in the dicot, which is a bean, please don't mention it that you're observing it or you saw it. Mention it when when you get to the monocot. Do you understand? So we're here to examine the bean seedling, look at the features it has and what function they would perform. All right, let's go. First column, the table, you write specimen D. The second column, you write seedling. What is the name of the seedling? You write bean seedling in bracket, a dicotyledon. Now, in case they ask you the next column, you should have what type of germination. You write Epigeal. In case they ask you why is the reason for choosing Epigeal germination, this is it. It's just like them asking you to define Epigeal germination. Now, you tell them that it undergoes Epigeal germination because the hypocotyle elongates. I will show that in the diagram. I will tell you the hypocotyle is the path below the cotyledon, but it is inside the soil. Now, when we brought this out of the soil, you can see it in the picture that the path below the cotyledon is the hypocotyle. It curves when it is growing, and it elongates to raise up the cotyledon above the soil. Do you understand? Hence, this is why it is called Epigeal germination. So, as I've explained Epigeal germination, why is it Epigeal germination? I repeat, it undergoes Epigeal germination because the hypocotyle, that is hypocotyle is spelled Hypocotyledon, COTYL. So the hypocotyl grows or elongates and raises the cotyledon, which is also called the seed leaf, above the soil. Now, once the cotyledon, this is why it is called epigeal germination. Then the next column, you have soil type. What kind of soil? The soil used in germinating this is, we tell them, loamy soil. Why? They may ask you, because one, it is dark, and it is a dark soil that is rich in what? Humus. It has high content of what? Humus. And what is humus? Those dead plant and animal remains that are found in the soil that act as what? Compost. So let's continue. Then the last column we have, let's go to that. Last column, you have, they may ask you 4 observable features of this young seedling. Because you are looking at a dicot, which is a bean, you write 1 under the observable features: presence of what? You tell them presence of or emergence of the radicle. Do you get? Presence of the radicle or emergence, meaning that the radicle has started coming out. What is radicle? In case you are confused, they may not ask you, but you need to know. A radicle is that path that grows into the roots, and it grows downwards into the soil, and it later forms the roots. Do you get? So remember, I say you tell them presence of radicle. Then the next one is still number 2 under observable features. You write presence of what? Plumule. Plumule is Plumule. Now, what is plumule? Plumule is that part that it is going to later grow into the shoot, to grow above the soil and come out of the soil and form the what? Shoot. So I've given you 2 observable features. Let's go to the next feature, number 3. Number 3 feature there is presence of the cotyledon, which was or you can write cotyledon, presence of a cotyledon which separates. Remember that the cotyledon will divide into 2, and it is from there the first foliage leaf, or the first foliage rather, will start coming out. The first leaf is called foliage. So when the cotyledon divides into 2, the leaf starts coming out from there. And as it continues growing, it forms the part we call the epicotyl. The epicotyl is mostly the part above the cotyledon. Remember, I told you that the hypocotyl is below the cotyledon. The epicotyl will later form other parts of the word shoots. Now, another one you can write, after writing presence of cotyledon that is separated, you write presence of what? Hypocotyl. So with this, I've given you at least 4 observable features of that what? Young seedling. Now, they may also ask you to state the same thing for the word specimen D, which is what? Guinea corn. So let's go to this in a tabular form. Specimen D, under the first column, you write specimen E rather, because we're talking about the next one, which is Guinea corn. The next column, after you've written the name on the first column, specimen E, the next column, they'll ask you seedling. What type of seedling? You write this. It is a Guinea corn seedling, in brackets monocotyledon. Then the next column will be a soil type. What soil did you use to grow it? Say it's a loamy soil, because loamy soil is the best soil for what? Growing crops, because of the high humus content. So you say, if they ask you what type of soil, you write loamy soil. Reason, if they ask you why, because it is dark in color and it contains high humus content. Then the next column, they will ask you for 4 or 3 observable features of the seedling of the Guinea corn plants. Now, number 1 feature, there are some things that there's some things you will see in the Guinea corn, which is a monocot, why it's growing, and you will never find it in the dicot. So let's look at these features. Just like I said, that there are some things you see in the young, or in the growing, or in the seedling of a Guinea corn, which is a monocot, you will not find in the seedling of a what? Dicot, like the bean. Number 1, we have presence of what? Instead of writing cotyledon, we now say presence of. Remember, okay, before I go into those observable features, remember that the maize plant, the Guinea corn, which is a monocot, will grow and come out of the soil. The first leaf or the foliage will be above the soil, but the cotyledon, which is the cotyledon which is found inside the Guinea corn seed that you threw into the soil to start growing or to start germinating, that Guinea corn seed will remain in the soil. It does not come up with the first foliage leaf. Unlike in the bean, which is a dicot, remember that the hypocotyledon extended and raised up that bean seedling above the soil. Do you see it? So this is a major difference you need to know. That is why it is not Epigeal. Just like you'll be seeing it in the diagrams or the images that I'm presenting to you, you will notice that in the one that grew above the soil, which is the Guinea corn, You only saw leaf. You did not see any guinea corn seedling that is decaying or rotting that grew out of the soil. It was only the leaf, the first foliage leaf or the first leaves that grew above the soil. But in the case of the bean, it did not even grow because it has not gotten to that stage where it can grow out of the soil because it's just 1 week. It's just a 1 week old seedling. But let's assume that it has stayed longer. When it now start coming out of the soil, it will raise up that bean that was inside the soil. That is what we will now call that's what we call the word cotyledon. That bean seed will be raised up above the soil, and it will come out with the help of the hypocotyl, which is stretching and elongating to raise it above the soil. You get now? Because of this type of germination, that is why I said in specimen E, under the seedling, you told them the seedling is guinea corn seedling. And if they ask you what type of germination, you tell them it is hypogeal. Now, if they ask you reason why it is hypogeal, you just simply define hypogeal germination. And what is that? What is hypogeal germination? It is the germination, it is a type of germination in which the cotyledon or the cotyledon remains inside the soil. It does not come out or grow above the soil. Meaning that it is not raised by the hypocotyl, but rather it is inside the soil. So if you are to define hypogeal germination, I repeat, is a germination in which the cotyledon remains inside the soil because the epicotyl doesn't stretch or elongate to raise it up. Are you following? The epicotyl only stretches, but it doesn't raise up the cotyledon. So that being said, that is it. Looking at the diagram, you see that that's the image of hypogeal germination in we could be in a maize, in a guinea corn, you get. But this is just hypogeal germination. So like I said, hypogeal germination is defined as that germination whereby the cotyledon remains below the soil. It remains inside the soil after germination, and then the next thing that follows is the elongation of the epicotyl. You understand? Why in Epigeal, the one that happens in beans, the cotyledon is carried above the soil. It comes out of the soil by the elongation, and this is done by the elongation of the hypocotyl. So do you understand? Now let us look at the last table, which I said that looking at 3 or 4 observable features of the seedling of the guinea corn. We want to look at some features we can find why a guinea corn seedling is growing. So number one, before we start, number one, let's go. Number one, we have presence of first foliage leaf above the soil. Then number 2, we have presence of cotyledon which remains inside the soil. Please always tell them where it is. Then number 3, we have presence of coleoptile which grows above the soil and it protects the plumule. I repeat again, that's number 3. Presence of the coleoptile. Coleoptile is spelled Coleoptile. Then I said it grows above the soil, that is, grows upward and it comes out of the soil. And what is the function of the coleoptile? It protects the plumule. And what is the plumule? The young shoots. That part of the plant that will later form the shoots. Do you get? Now that means we move to the next one. That should be number 4. Presence of coleorhiza. Now what is coleorhiza? Coleorhiza is, before I explain, it is spelled Coleorhiza. Now the coleorhiza is opposite of the coleoptile. Just like the coleoptile is protecting the plumule that will later form the shoots, the coleorhiza protects the radicle that will later form the roots. That is why the coleorhiza grows downwards into the soil. So this is another feature. Now the last feature I'll give you is presence of the scutellum. Now what is a scutellum? Scutellum, before I explain, it is spelled SCUTELL, that is LLUM. The scutellum is what we call cotyledon in monocot. we don't use the word cotyledon, we call it scutellum, but in dicot we call it cotyledon, but they are the same thing. So what we said, another point is presence of scutellum. The scutellum is the cotyledon in monocot, and it grows or remains inside the soil. It does not come up with the word foliage leaf or with the hypocotyl, unlike in the case of the dicot where it does this. So with this, I've given you some features, and also you can use this as differences between specimen Z, which is a young a potted bean seedling, and specimen E, a Guinea corn seedling, a potted Guinea corn seedling. So this is a very important point you need to know in this for this specimen and how to answer this question. So with this, I'm very sure that any question coming from specimen Z, which is the word potted bean seedling, or specimen E, the potted Guinea corn seedling, you should be able to answer it perfectly well and earn your mark. Thanks for sticking to this channel, Calista's Bio Classroom. Stay tuned for other detailed information on other Neco specimen, as I'll be bringing them on before the exam, which is next week Wednesday. Bye.